distinguishing programs underpinning disease tolerance and resistance against infections.
So can you hear me now?
Yeah, I do.
Great. So can you see my presentation?
Yes.
Great. So first of all, I would like to apologize for not being able to present. It was just a problem with my documents in the entrance to the US. I will talk about disease tolerance and resistance, mostly in influenza virus infection. It's a joint collaboration with Eran Bachach from Tel Aviv University. He's a virologist, and the work was done by Ophir Cohen and Gal, PhD students in my lab. Our main interest when we started the project was to understand more about why different individuals show a distinct disease outcome. We decided to do some design that will provide us information, as much information as we can. So we started with CC mice. It's a collection of mice. Each of them is an inbred strain, and each one is a different strain, and you can take as many as you want from each strain. So it's a bit different from other projects, usually in mice that most researchers are using only one strain. Then with many strains, we can see the variation in the population. And we did in vivo influenza infection. We did several time points, 5 time points, and also the no infection. And we took many different clinical parameters and also gene expression from the lungs. So it's different from human studies where the lung tissue is not available. And also we can take early time points that are not available in infections in human studies. So overall, we have about 250 samples that were collected, and we worked with this data. So just about the clinical data. So for example, we have the weight loss. Weight loss is very informative in mice. Also, we have measurements of the breathing dysfunctions, cytokines, the damage of the lungs, the cilia, and so on, and the messenger RNA profiling. So for example, here we can see over time points for the entire population. We can see increase in the viral burden, in the weight loss, breathing, and the tissue damage over time. And if you focus, for example, on one specific time point for the weight loss, for example, and here is in the right is the extension for across all CC mouse strains, the weight loss, we can see that each strain has a different weight loss. So for example, some of the strains here on the right, they lose almost 25% of the weight after only 4 days, right? So they're very, very sensitive, and the others, like the ones that are at the left, are just continue to gain weight. Although we know that they are infected, we see the virus in them, but they are continue to gain weight. So we have this variation, and also the standard variation is quite small. So it seems as a genetic effect, and the clean data. So we continue to work on this. But one more thing to say just about this variation. So first we can look at the variation in the phenotype, in the clinical phenotype as the weight loss. But in addition, I can compare the tissue damage or the weight loss or any other clinical phenotype here in the y-axis to the viral burden, to the quantity of the virus, the concentration of the virus in the lungs, in the x-axis. And here, each point here is a different individual mouse. And what we can see is that there is a wide variation, for example, here in the tissue damage in the cilia of the lung, for the same viral burden. So for the same amount of virus, I see very different levels of tissue damage. And this we call disease tolerance, meaning that for the same level of infection, I see different levels of susceptibility of the host. The ability of the host to tolerate or to maintain the health under the same level of the virus. So we see both levels of variation, both in the clinical data itself and also in the relations between the health and the viral burden, the disease tolerance. So we are satisfied with this because this opens the possibility that we can see maybe the molecular basis for this variation. Okay, so now I'm just briefly talk about our approach. So the first thing that we did is to do dimension reduction. There is two options to do dimension reduction, either to reduce the dimensionality of the individuals or to reduce the dimensionality of the genes. And I'm focusing now only on the gene expression data, just the gene expression data from the 250 individuals. So I'm taking all individuals from all time points together, from all strands together, everything together, just to consider it as variation. And we did dimension reduction so that in the two-dimensional map, each point is a gene. After we consider all the data together, we use the autoencoder neural network, but it can be also other ways to do dimension reduction. This worked the best. So the final map that we have is a map of a similarity rule. It's a dimension reduction. So each two genes have the same, maybe very similar behavior across all time points and across all strains, or all mouse strains. So they are really similar in many aspects, not only in specific strain or in specific time point. So now the next step is just to look in each specific individual. Here, each map is one specific individual, and the positions of the map are the same always. We just color coding the measurements of each individual in its own map. And we see that some individuals have for example, here, individual number 1, the direction is to the top left from blue to red. And for this individual 3 at the right here, we see the direction from the bottom left to the top right. So each one is a different overall state in this map. So to analyze this quantitatively, we did a deconvolution in order to get the state of each individual on the y-axis and on the x-axis. And this is the map in the right here. We have in the x-axis and in the y-axis, we see the states, the deconvolved states based on this map of each individual. So each individual is a point, and for example, individual 3, the direction for both the x and y axis is positive. So we see it in the positive in both the x and y axis. We call this two axes as T and R for the x and y axis, and I will explain shortly why we did it. So it's the T level and the R level of each individual. It's just 2 scores for each individual based on the dimension reduction. Actually, we could do the opposite. Maybe we could do the opposite to reduce the dimension first to get the map of the individuals and then to create the map of the genes, but it's not a linear dimension reduction and we don't get the same results. Okay, only if we first reduce the dimension of the genes and then the dimension of the states of the individuals, we get this solution. So what's special about this work is that we spent a lot of time in trying to understand what are these two main axes, because we saw, and I do not show it here, but we saw that the two axes explain a very large percentage of the variation and very similarly explain a large percentage of the variation. So they should be very interesting for infections. So just to go to what maybe essentially we got. So first, both of these levels, T and R levels across the population, are increasing during infection. We see them almost always increasing together during infection. Both of them are increasing in many different infections that we tested, also increasing in wound healing. But if we look at maybe the trivial splits that we could think about, for example, if maybe one of them is innate and the other is adaptive, or maybe one of them is pro-inflammation and the other is anti-inflammation. But in all different aspects that are known, we saw that both of them are the same. So it was not a trivial explanation for these two distinct programs. So we thought maybe it would be related to resistance and disease tolerance. I mentioned it briefly, I'll explain it again. Resistance is a program of pathogen elimination, just the molecular program that fights to eliminate the pathogen. And the disease tolerance is it's more a physiological property of the maintenance of the health in the presence of a pathogen. So it's the relationship between the health and the amount of the virus here, the pathogen. So that's disease tolerance, and disease tolerance is not defined. First, it's not immune tolerance, the lack of responsiveness to antigen. It's disease tolerance. And it's not defined molecularly, it's just defined physiologically by the relations between the two physiological parameters in the literature. And what we've shown is that we have three reasons to believe that program R, the first program, the y-axis, is related to resistance ability, the ability of the host to eliminate the pathogen, and program T is related or associated with disease tolerance, the ability to maintain health in the presence of the pathogen. So I will start with the first evidence. We did functional analysis. So for example, here is the map. Each point here is an individual, and we can do color coding by the level of specific genes, for example, ROC1 or TRAF1, and we see some directions. It's endotypes based on specific genes. So based on this map, we can characterize each gene for its relations with the T program and the R program. For example, ROC1 is negatively correlated with the x-axis, which is the T program, so it's negatively correlated with the T program, and so on. So based on these associations of each gene with the T and R programs, we can do functional analysis. And this is the results of the functional analysis. For in the y-axis, it's the enrichments, functional enrichment in program R. X-axis is functional enrichments in program T. And surprisingly, what we saw that for program R, we see many functions related to resistance. For example, cytokine storms, PR signalings, antigen presentation, all of them are against the pathogen. But when we look at the x-axis in the enrichment in the program T, we see wound healing and ECM and angiogenesis, many functions related to maintaining the health of the host. It's not against the virus, it's more for the health of the host. So that's the first evidence contributing to the idea that that's disease tolerance and not resistance, the T program. Now for evidence 2. Evidence 2 relates to the physiological phenotype, because disease tolerance and resistance are actually defined at the physiological level, or previously were defined at the physiological level. So we correlated the T and the R level with the viral burden and also with the relationships between the host health and the amount of the virus, which is the disease tolerance signature at the physiological level. And what we saw that the R levels are correlated with the viral burden. That's a signature hallmark of resistance. But the T level correlated with the disease tolerance phenotype, the ratio of tissue damage to the viral burden. So we have at the phenotypic level, across the cohort, we see the hallmarks of resistance and the hallmarks of tolerance for the R and T levels respectively. And the third evidence is just that we took many functional classes of response to different stress, different types of stress, So we split them into biotic stress and abiotic stress. If it's any kind of pathogen, it's biotic stress, and otherwise it's abiotic stress. And then we do, and again, enrichment analysis, and we see that for the x-axis, which is the program T, we see association with biotic and abiotic stress here in 2 colors. But for the program R, the resistance program, we see associations only with the biotic stress. And this is in accordance with the general understanding that if it's to maintain the health, you need it both for biotic and abiotic stress. But if it's to fight against a pathogen, you need it only for the resistance program. So this also supports, based on analysis across many different types of stress, that we see here, indeed, the program to maintain health during infection and the program to resist against the virus. So we did it in bulk tissue, bulk lungs, but then we tested it in many different other data sets. We see the same signatures retrospectively in bronchial epithelial cells in human and many other immune cell types in human individuals across cohorts. So we see the same signatures if we know what to look. We see the same signatures. So we think that it's first, it's intracellular cell autonomous states. It's not only at the level of the bulk tissue. And we think that it's both for immune cells and non-immune cells, both innate and adaptive immune cells. So the general programs, just like for example, PRRs and TLR signaling is a general programs, interferon signaling is a general. Also, the split for these 2 programs is a general split between 2 distinct states in response to infection, usually responding together. But since we had different genetic backgrounds, could use the fact that they are not always synchronized completely, and then we could split them. So just briefly, I will mention here that we did a lot of experimental work. What we did is to do associations of genes to each of these programs across many different data sets, infections and autoimmune disease, to see which are the specific genes that most correlated with each of these programs. And this you see here the ranking for the T and R programs. And for the resistance, it's easy. We found the top ranked ones were just many of them just interferon signaling and genes that are known to be validated IV restriction factors to influenza. So it makes sense. But for the x-axis, it's less defined, because usually today the disease tolerance is not defined at the molecular level. So there are no known regulators of disease tolerance. So we saw these 2 ones that the Argadia and MAP2K2 that seems very strong one. We chose the Argadia and we validated, I did not show it here, but we validated that in vitro, that if we do perturbation for Argadia, we perturb specifically the T program, not the R program, both for overexpression and for perturbation of it. And also we affect the disease tolerance phenotype, so meaning the ratio between the cell death and the amount of the virus per cell. So that's for validation, or maybe putative regulator for the disease tolerance. And also, if we are trying to see if these programs can predict phenotypic diversity, so we look at the levels of the T and R programs before infection, and then we look at different phenotypes of severity of infection, of course, after the infection. So the relationships between the T levels before the infection and the disease phenotype came very strong for the CC mice, for example. We see the T levels before infection in lungs correlated with the severity of infection. Then we validated this also in another cohort of mice, the BXD cohort, that's published data. It's the same results. And also in human blood, if you take macrophages in human blood, T levels against the severity of infection, again, we see the same relationships. And we don't see these relationships with the resistant programs. So we think that disease tolerance have an effect, specific effect on the severity of infection rather than resistance, which might be quite surprising. So this paper is already in the bioarchive, and Gal and Ophir led this project together with our collaborator Ran, and many others helped with this interpretation, including Fouad Iraqi for the CC mice, Michal Netia and Michal Mendelbaum, Nira Cohen, Ido Amit for the interpretation of the results. So thank you all for your attention, and I will be glad to take questions.
So it's very interesting, and I was wondering if you've been thinking about, how this disease tolerance versus, response could affect people's response to immunization. We see that sometimes people who are immunized, they have really strong response, while others, don't feel anything. So would that distinction be predictive of the response to immunization?
Yes, that's a great question. So we thought the same. We just started a collaboration to investigate actually trained immunity, just what would be the long-term consequence of the vaccination, and we do it with yellow fever virus and with BCG specifically. So we don't know, we just started now.
Okay, so we've been thinking about it. Well, nice.
Yeah, we collected the data, and we started to investigate this. Because you need enough individuals and to have control data to investigate this.
Hi, Irit, great talk. Can you go back to the slide where you have this human example? One more. Yeah. So what are no?
This one.
This one. Yeah. So in the human patient, what do the points mean? In the left, it looks like they mean individual mice, but on the left, what do they mean?
Can you repeat the question? What left?
What do we see on the human patient example on the right? What is the meaning points that we see?
Okay, so this data set is gene expression data set. It's a very large data set in which they took blood of individuals at the beginning of the winter and before any infection in the winter, and then if they were sick, they came and then they tested if they had influenza, then they took gene expression after during disease. So what we had is 2 days post symptoms here, and also the blood gene expression before infection about 3 months before the infection. That's what we have here. It's not just at the beginning of the infection. And so they did macrophages. So for the x-axis, we calculated T level for the blood MFs, and for the y-axis, we didn't have clinical data for these individuals. So we took only the R level at 2 days post symptoms, because we know that the R level is correlated very well with the amount of the virus, for example. And here I just show the amount of the virus, but actually if I look at all other symptoms in mice, we see the very good correlations with the R levels and the symptoms. So we know that it's a good indication, the R level, for the severity of disease.
Okay, thanks. Great.
Anyone else? Okay, thank you, Irit.
Thanks so much.
Wonderful. Thank you.